Hello, everyone. Welcome to the ninth episode of the Naked Immunology series webinar series. My name is Nicolas Gaudenzio. I'm a scientist at the French NIH. I am the CSO at Genoskin. And today it's my great pleasure to welcome Etienne Meunier, the Dr. Etienne Meunier from France, from Toulouse as well. So a little bit of intro on Etienne. He obtained his PhD in 2013, where he did in Toulouse in the Bernard Pippi lab in France. He then moved for a postdoctoral study, a very productive postdoctoral in Basel in Switzerland, where he worked in the lab of Professor Peter Bross and published two important papers in Nature and Nature Immunology, working on bacterial sensing and inflammasome in the context of cell autonomous immunity. In 2017, Etienne moved back to Toulouse at the IPBS Institute, where he obtained a prestigious funding from the European Research Council. He created his own lab, and now his group's really pioneer studies on NLRP1 inflammasome and epithelial immunity. And not stopping there, his research has been recognized by the CNRS. He received the bronze medal that recognized one of the most outstanding researcher in France, actually, and he has been elected with a strong future potential for research. So today, Etienne, it's an honor to host you today, and I will give you the floor for the presentation. So everyone, just if you have any question, feel free to ask them in the chat, and at the end of the presentation, I will share the question with Etienne, and then he will be able to respond in live, so we can make this as much interactive as we can. Thank you, everyone, and have a good webinar.
Okay, thanks, Nico, and hi, everyone. So it's a great pleasure to be there today. And what I would like to share with you indeed is something around a specific inflammasome complex, which is an innate immune sensor called the NLRP1 inflammasome, directly at mediating host immune response in the bordering epithelial cells. So in general, my group studies specifically cell death processes and the way those cell death processes will influence the induction or not of the immune response. So we base our research on the fact that there are thousands and thousands of cell death events every day in human beings, which has brought Lewis Thomas to say that by the time he was born, more of him had died and survived. And the central question of the lab is how do cell death processes are occurring and what for? So there is a huge sigma catalog of cell deaths actually, starting from 1842 to 1972, where apoptosis, an anti-inflammatory form of cell death, has been described. But the first observation actually was done by Carl Vogt in the University of Geneva in Toulouse. And you can see that among the years, plenty of weird and different cell death processes that are regulated and encoded genetically have been described. In the lab, we particularly study one cell death process that is apoptosis. And it's clear that each cell death will have a specific function. As you can see, apoptosis is a very inflammatory form of cell death, necrotic, that will bring some different response than apoptosis. When you see an apoptotic cell going on, it makes blebs, compact everything inside, and do not freeze everything. So when we speak about apoptosis that has been discovered in 2001, is that there is a huge and intense literature that describes its role in various contexts as a host protection or when dysregulated as driving pathology. One way of inducing apoptosis is through the engagement of these intracellular and pro-inflammatory complexes that we call the inflammasome. And an inflammasome is composed of a sensor, an adaptor protein, and a protease, caspase-1 here, or the non-canonical inflammasome with caspase-4 and 5 in human. And once an inflammasome complex can get activated, it activates the caspase-1 that will have two functions. First, it will process, cleave, and mature the IL-1 cytokines, such as IL-1 beta, L18, and the protein gazamin D. And the cleaved gazamin D will form a pore at the plasma membrane, ensuring two things. First, the release of the mature form of IL-1 beta and L18 that will act at the communication level by promoting the neutrophil recruitment, the process of fever, the production of gamma interferon, for instance. But also, gazamine D will promote pyroptosis, the pro-inflammatory form of cell death, which has been shown to be extremely important at restricting a broad range of different pathogens. So given the strength of the inflammasome, it's not surprising that beyond humans and rodents model that we currently use, several species have developed several inflammasome or inflammasome-like responses that range from plant to human. And in the broad range of inflammasome catalog, you can find, according to the different species, a lot of different receptors that can form an inflammasome and respond to a broad range of signal. Here we will focus on the human NLRP1 inflammasome. But why the human NLRP1 inflammasome? First, it was the first inflammasome discovered by Fabio Martino and colleagues in 2002. But it took almost 15 to 16 years to the scientists to understand why and what the human NLRP1 inflammasome is doing and responding to. Indeed, when we look on the NLRP1 inflammasome, most of our studies have been performed in rodent models that are extremely valuable models, but that show strong divergences in terms of structural and functional function of the inflammasome. And this is particularly the case for NLRP1. For instance, in human, the NLRP1 inflammasome is located in the skin, mouth, tongue keratinocyte, in airway epithelial cells, in corneal epithelial cells, in some subset of colon epithelial cells, endothelial cells, and maybe in neurons. In rodent species, the NLRP1 from those species is expressed in macrophages, monocytes, and neutrophils mostly, which are professional immune cells. This is the first difference. But the second one was that actually NLRP1 has been selected according to the pressure each host has. So if we take, for instance, the rodent, it has been shown that the bacteria Bacillus anthracis, that is responsible for the anthrax present in the soil, or the parasites Leishmania and Toxoplasma gondii, are robust activators of the rodent NLRP1 inflammasome. But then when you transfer those pathogens into the human NLRP1, you don't get any activation. And for 15 years, it has been struggling many scientists, including myself. Until the laboratory of Bruno Reversad and the work from Franklin Zhang could show that gain of function mutation in humans for NLRP1 inflammasome were existing and driving pathologies. And as you can see, they could show that at the skin, the eye, and the foot of patients, you have this kind of necrosis and several profile that occur. And this was mostly due to gain of function mutation in NLRP1, human NLRP1 inflammasome. So many research then has been addressed to characterize various different gain of function mutation, and I just bring you in all those labs, fantastic labs that could highlight this process. But this has opened the question and the part of the answer that NLRP1 in humans actually could form an inflammasome. We are just missing about the activating signal and the signaling pathways. The subsequent work from the Franklin lab actually, and from different other labs such as Hans Dietmar in Switzerland, could show that one environmental trigger in humans, the UVB that are released by the sun, could actually induce the activation of the human NLRP1 inflammasome in the skin, but not in rodent model. And this was occurring through a process where the RNA were cross-linked and altered by the UVB, leading to the translation of protein inactivation. Then ribosome would stall and recruit one MAP kinases, one stress kinase that is called ZAC-alpha, that will autophosphorylate and induce a signaling pathway called the ribotoxic stress response. That will end with the P38 stress kinase phosphorylation, the phosphorylation of the human NLRP1 inflammasome, and activation. And this occurs in basal keratinocytes, but not in fibroblasts, melanocytes, professional immune cells, including the mast cells. We don't know about the nerves, but also according to the type of trigger in some human endothelial cells. And the difference between human and rodent response in terms of NLRP1 inflammasome is due to the fact that in human, we have this small region that is called a disordered region that is highly phosphorylated by the P38 and ZAC alpha kinases, whereas rodents and NLRP1 lack this region. So this observation encouraged me to address the role of the NLRP1 inflammasome, and mostly during infectious diseases. And to do so, we started with the observation of infection by the opportunistic pathogen Pseudomonas aeruginosa. Indeed, this pathogen is able to infect a broad range of sites that are important for NLRP1 response, the airway, the cornea, and the skin. And it affects mostly immunodepressed patients and people carrying genetic diseases such as cystic fibrosis or celiac dyskinesia. In addition, it has been well known from the medical side that epithelial cells from patients release IL-1 cytokines and exhibit cell damages in chronic infection with Pseudomonas. Yet in vitro results could never reproduce such a phenotype. So here, given this clinical status, we hypothesize that as IL-1 cytokines and cell damages might rely on the inflammasome pathway, that might NLRP1 could play a role in response to Pseudomonas. So to mimic a chronic infection by Pseudomonas aeruginosa, which is an extracellular pathogen, we use an inverted transwell method. Usually people put the cells at the top of the transwell. Here we did put primary human corneal epithelial cells from the eye or primary human nasal epithelial cells. And we did put them at the bottom of the well and put Pseudomonas at the top to see if some release factor from chronic like infection by Pseudomonas might induce such a pathway in those primary cells. So, and you can see here that in human nasal cells, there is in presence of Pseudomonas IL-1 beta release. The same is true for corneal cells in black. And if you use the ZYVAD or the pan-caspase inhibitor ZVAD, you lose IL-1 beta release, which suggested to us that an inflammasome indeed was activated in response to extracellular Pseudomonas infection. So to determine which inflammasome was expressed, we took all inflammasome sensors known to be expressed in human and used their ability to oligomerize when they are activated to form what we call a spec. So basically, we did put the adapter protein with the GFP tag in rapporteur cells, and then add an NLRP1, an NLRP10, or IM2 inflammasome sensor individually in those cells. If an inflammasome is not activated, you will have a diffuse ASC fluorescence. If an inflammasome is activated, you will have a single spot that really show that there is something triggering this specific inflammasome. To make it short, we can validate here that our cells, in blue is the TAPI, in green is ASCGFP, without stimulation or without inflammasome expression, have a broad diffuse ASC fluorescence. But then, if you add an NLRP1 here, without stimulation, you don't have anything, but with stimulation, you can clearly see that there is an active inflammasome per cell. And this showed to us that NLRP1 might be a sensor to Pseudomonas aeruginosa. So to determine which factor from Pseudomonas could activate NLRP1, we made a screen on the bacterial mutants to see which toxin or which element might trigger such a process. And to go directly into the result, what we could show is that the type 2 secretion system for Pseudomonas, that is a nanomachine able to inject in the extracellular environment a lot of toxins, was important as no spec was found in our cells when infected with Pseudomonas lacking the type 2 secretion system. And in this type 2 secretion system, we have 4 major virulence factors that are released, including the exotoxin A. And we depleted each one of them and observed the ability of our reporter cells to form active inflammasome. And as you can see, when you deplete the exotoxin A, you lose completely the formation of the NLRP1 inflammasome, characterized by the bright spot. And exotoxin A for us is a very interesting target. Indeed, it shares a similar activity than 2 other virulence factors that are the diphtheria toxin and Colix toxin. they have the ability to ADP ribosylate an essential gene and protein, EF2, involved in the regulation of the translation machinery. And the way they do is that they target a specific amino acid expressed only by EF2 that is called the diptamide. So diptamide is a transformation from the histidine that is catalyzed by all these DPH enzyme in order to form a diptamide. And this leads to a ribosome stalling and collision. So as EF2 is an essential gene, we decided to remove the DPH enzyme. So we removed the DPH1 enzyme and evaluated the ability of exotoxin A to activate the NLRP1 inflammasome in NLRP1 reporter cell lines or in NLRP1 DPH1 knockout cell lines. And as you can see, you have the bright spots that appear with exotoxin A and they completely disappear in DPH1 knockout cells. The same is true for diphtheria and colic toxin that both induce the same process than the exotoxin A. And then if you take a control for the NLRP1 inflammasome activation, which is the Valboro Pro, able to trigger NLRP1 inflammasome response without the need of the ribotoxic stress response, you don't have a difference between both cell lines. So relying on these findings and the findings from the Zhang lab previously, we could show that Pseudomonas exotoxin A, by ribosylating EF2, is able to block the translation, promote ribosome collision, and we wanted to know next if this collision was able to trigger a ZAC-alpha dependent P38 activation of the NLRP1 inflammasome in human. So we generated ZAC-alpha knockout reporter cells and exposed them to exotoxin A. So I invite you just to look at the red squares in this NDGest biochemistry approach. And what you can see here, when you expose your cells to exotoxin A, GNK, and P38 that are downstream to ZAC-alpha are phosphorylated, which means that they are activated. In the ZAC knockout, you lose completely this phosphorylation, suggesting that ZAC was important. But at this step, we need the genetic. So we generated ZAC-alpha knockout cells in our reporter cells. And as you can see, when you expose your cells, your wild-type cells make the specks. When you expose the ZAC-alpha knockout cells to Pseudomonas or exotoxin A, all of a sudden you lose everything, showing that ZAC-alpha indeed was activated by the exotoxin A from Pseudomonas to promote NLRP1 activation. But we all know that downstream of ZAC-alpha, you have three major MAP kinases, GNK, P38, and ERK. To make it short, we did invalidate all of them and proved that the P38 alpha and beta knockout was able to completely inhibit exotoxin A-induced NLRP1 inflammasome response. So at this step, we have Pseudomonas exotoxin A that actually activates the human NLRP1 inflammasome by blocking the ribosome, inducing ZAC-alpha, P38, and NLRP1 activation. Next, we aimed to go back to a bit of clinic. We know that Pseudomonas loves patients that exhibit cystic fibrosis disease. And cystic fibrosis is a monogenic disease where the CFTR, the chloride channel, has been mutated or is unable to be expressed in various cell types, including epithelial, airway epithelial cells. So we exposed, thanks to the collaboration with the Hospital of Toulouse, human nasal cells to exotoxin A or cystic fibrosis human nasal cells to the same toxin, and observed, surprisingly, that those cystic fibrosis cells were able to die more potently and faster than the wild-type cells. And this was fully ZAC-alpha dependent, as the inhibition of ZAC-alpha completely reverted this process. So aiming to know if ZAC-alpha was dysregulated in such a process, we perform a phos-tag. So actually it's a migration delay if the protein is phosphorylated. And you can see that in healthy cells, exo-A induces phosphorylation of ZAC-alpha, the switch to the top. And if you put the PLX, the inhibitor of ZAC-alpha, it gets down. But then in cystic fibrosis cells, we are surprised to see that the level of phosphorylated ZAC-alpha were the same, suggesting that this pathway was not dysregulating in such a context. But what we could observe is that actually P38, phosphorylated P38 that occurs downstream of ZAC-alpha was completely dysregulated, even from the beginning of the exposure. You can see here that by measuring phosphorylated P38, in wild type you have an accumulation of phosphorylated P38, and then it decreases over time. In the cystic fibrosis cells exposed to exotoxin A, you can see that phosphorylation of P38 is already present at T0 and maintained over time, suggesting that actually dysregulated P38 was probably the rationale why the cystic fibrosis cells were responding higher to exotoxin A. So to answer this question, what we did is that we used the novel therapy of cystic fibrosis and exposed our cystic fibrosis cells to Trikafta, this therapy that allows the re-expression and the readdressing of the CFTR at the plasma membrane, and expose them to exotoxin A, or in presence or absence of the ZAC alpha inhibitor. What we can see is that cystic fibrosis cells, they extensively die in response to exotoxin A, and that pretreatment with the Trikafta for 3 weeks actually completely reverted this process. And the cell death level observed in Trikafta-treated cells were actually superposing the cell death induced by exotoxin A in wild-type cells. And more interestingly is that no matter the type of cells you have, if you use the PLX inhibitor that inhibits ZAC alpha, you revert completely the cell death. So to make this story short that has already been published, we could prove that Pseudomonas, but also the diphtheria and some subspecies of Vibrio cholera actually all activate the human NLRP1 inflammasome, but not the rodent NLRP1 inflammasome. In Pseudomonas, this is type 2 secretion secreted toxin driven thanks to EXO-A triggered EF2 inactivation. And this leads to ribosome collision, ZAC alpha inactivation, P38 dependent human NLRP1 phosphorylation and activation. We could also show that cystic fibrosis cells are more sensitive to EXO-A driven response, and that this is probably due to P38 dysregulation in those cells. And finally, we could also highlight that the therapy, the Trikafta therapy, is able to reverse the CF sensitivity to exotoxin A. But now I would like to share with you a very recent study that we did on the NLRP1 inflammasome that does not directly rely on infectious diseases, and where we performed a very complementary and worldwide detective approach to understand some novel and emerging dermatitis that occur in various fishermen all around the world. So we got contacted a few years ago about some fishermen in Senegal, actually, that exhibited after fishing some dermatitis and necrosis of the skin, as you can see here. And investigation could rule out any infectious origin or any pollution origin. And this has been a strong panic for the fishermen and their families, because they could not at least go fishing and could not live about their work. So when we were contacted by Philippe Hess from Ifremer and Loic, they told us that they could gather some samples from the fishing boat, the net, and the different environmental places. So we asked them to give us the different fractions that they could isolate, and decided to test those fractions. What they could show first is that in the water, there was one microalgae that I never heard about that is called Vulcanodinium rugosum. And the second thing that I could identify in the samples were 2 major toxins, pinotoxins and portinin, but none of them have been linked to any kind of dermatitis or any kind of strong pathological disease, inflammatory disease. So we decided to investigate this aspect. So we took the different samples from the boat fishermen and exposed primary human keratinocytes to those samples, and performed a very broad analysis of the type of inflammation or inflammatory response those keratinocytes could exhibit. We found a very restricted type of response from those keratinocytes characterized by IL-1 beta, IL-1 ERA, and IL-18, IL-1 alpha release, only cytokines that rely on inflammasome signaling. The same was true when we tried to see if those samples were toxic. When we addressed the toxicity of the samples on keratinocyte, you can see that if you use the caspase inhibitor or the caspase-1 inhibitor here, you inhibit the cell lysis induced by the samples on keratinocyte. And we could confirm also a strong release of IL-1 beta, IL-1 alpha, and IL-18 that was caspase dependent in keratinocyte in response to those blood samples. So when we say caspase, IL-1 beta, and cell death, we immediately think about inflammasome signaling. So before addressing the inflammasome signaling, we could benefit from the Ifremer samples to the fact that they identified two types of toxins in those samples: portimine, namely portimine A, and pinatoxin G and H. So we purified those toxins and exposed individual keratinocytes to individually purified toxin. And you can see that in black, only portimine was able to induce cell lysis, IL-1 beta release, and IL-18 release. Pinatoxin had no effect, even with scandalous amounts on the cells. So we decided to investigate if there was an inflammasome activated by portimine that might actually explain the dermatitis observed in the fisherman. So to do so, we use the same approach than previously described. We use our reporter cell lines that express all the keratinocyte sensors individually and expose them to purified portimine or sample from the pot. To make it short, again, we could find that NLRP1 with the bright specks was activated in response to the samples, but not NLRP3, NLRP10, or AM2 inflammasomes. So in order to validate those findings, we invalidated in primary keratinocyte NLRP1 by CRISPR-Cas9 and measured their ability to release IL-1 beta and to undergo cell lysis in response to the sample from the pot, purified portimine, and two activators of the NLRP1 inflammasome, the valboro and the anisomycin. And as you can see, sample 2 or portimine-induced cell death is partly abrogated, and IL-1 beta release is completely abrogated in the NLRP1 deficient keratinocyte exposed to sample 2 and portimine A. So in order to go further into the mechanistic of such a process, we aimed at determining how portimine was able to induce NLRP1 inflammasome activation. So we hypothesized that portimine might actually be a ribotoxin from the environmental environment driving such a pathway, ZAC-alpha, P38, NLRP1 phosphorylation. So first, to determine if portimine is able to inhibit translation, we made a simple test. We measured the incorporation of puromycin into the nascent peptide chain of the ribosome. If the ribosome is working well, puromycin will be incorporated, and with a Western blot, you can see a smear. If translation is not working anymore, it's a gray zone. So you can see that in a mock, everything is going fine. In sample 2 or portimine exposed samples, there is no translation anymore. Everything is gone, suggesting that indeed portimine is able to block translation through a yet to be determined mechanism. To go further, we followed the same principle than for exotoxin A. We could show that portimine A and sample 2 are able to activate phosphorylation of ZAC by phosphotype. And when removing ZAC-alpha, phosphorylation of P38 induced by portimine A was completely gone, suggesting that portimine activates the same pathway than exotoxin A, the ZAC-alpha driven P38 and NLRP1 response. So we generated ZAC-alpha knockout primary keratinocyte and measured the ability to release IL-1 beta and undergo cell death by determining the percentage of cytoxin incorporation. And you can see that portimine induced beautiful IL-1 beta release, which is completely gone in the ZAC-alpha knockout. The same is true for the cytox incorporation. So then we decided to go further. As I mentioned at the beginning, the major difference between the rodent NLRP1 and the human NLRP1 is that in this specific domain, the human NLRP1 is highly sensitive to phosphorylation. And 3 major sites have been found by different teams as being sensitive to P38-driven phosphorylation. The serine 107, the serine threonine 112 to 14, and the 178 and to 180 TST serine and threonine. So first, we wanted to know if P38 activated by portimine was able to phosphorylate NLRP1. What we did is that, thanks to the help of Franklin Zhang, we could express this specific domain coupled to a snap tag to measure the ability of portimine to induce phosphorylation of this domain in NLRP1. And by phosphotype, you can see here the NLRP1 disordered region, which is this region. When it is not exposed to anything, you have this kind of pattern of phosphorylation, meaning that probably the endogenous NLRP1 is already phosphorylated in steady state. But when you add increasing amount of portimine, you see a huge switch between this low phosphorylation status of NLRP1 disorder region to highly phosphorylated NLRP1 disorder. We could also validate that ZAC alpha was very well activated by portimine as P38 was. So then when we use the inhibitor of P38, you can see that the other spikes induced by portimine were gone in our reporter cells. But what does it mean? It means that these phosphorylation sites are important to drive specifically NLRP1 inflammasome activation in response to portimine, but probably other ribotoxic stressors. And then we came back to genetic. We found that in the genome ID sequence database that some humans actually are polymorphic for those serine and threonine that seem to be important for portimine-induced NLRP1 inflammasome. So we expressed those variants into reporter cells and measured the ability of portimine to induce the activation of those NLRP1 variants. As you can see, the wild-type NLRP1 respond well to portimine, make the specs. But if you take the variants, the S100F or the DEL P108 threonine 112, you don't have spec anymore. And this is easily quantified and robust, suggesting that naturally occurring mutation in NLRP1 abrogate portimine by probably other ribotoxic stressor induce inflammasome activation. So to resume this study, we could show that portimine induce a ribotoxic stress response, that it drives the hyperphosphorylation of ZAC and P38, and this activated kinase will phosphorylate NLRP1. Portimine induces the secretion of the pro-inflammatory cytokine and induce pyroptosis in an NLRP1 dependent manner, and we are not equals. Some people might actually resist portimine exposure thanks to NLRP1 polymorphism. So then, having this biochemical knowledge, we wanted to get back to this kind of physiopathological field and to see whether portimine was actually the trigger or able to induce the similar dermatitis as observed in the fisherman. So to do so, the lab of Franklin and John Common in Singapore generated organotypic skin from keratinocytes in which they genetically invalidate ZAC alpha. They exposed this organotype to portimine A and analyzed the response of this organoid. As you can see, portimine in wild-type cells will induce a detachment of the epidermidis, a lot of keratinocyte death and condensate, a process that is partly reverted in the ZAC alpha knockout organoids. The same is true. What they did is that they analyzed also the full inflammatory profile from those keratinocytes. And could find that actually among all the hits observed induced by portimine, IL-1 alpha, IL-1 beta, and IL-18 were among the major hits released by this organotypic skin in a ZAC alpha dependent manner. As I mentioned at the beginning, in rodent for now, we cannot show if the NLRP1 response can drive a pathology and inflammatory response as it is not concerned. And so far, we have failed to humanize these rodent models. But for a reason that we are unable for now to explain, the zebrafish NLRP1 seem to be closer to the human NLRP1 than to rodent. So what we did is that we went to Spain with Fran Roca and Miriam Pinilla, who are working on zebrafish, and asked them if we could test the portimine effect on zebrafish. What they could show is that the portimine is highly toxic at 25 nanomolar, which is nothing compared to what people found in the net sample from the fishermen on the zebrafish. So they analyzed on the fin-tailed damage on the skin of zebrafish by ranking them to normal. Here the skin is fine, to moderate, or to highly necrotic here with the dark, very dark and retracted tail. What they could show is that actually portimine induces a huge cell death in the skin of zebrafish. And that this cell death, if you remove ZAC alpha in the zebrafish or NLRP1, is completely reverted in the ZAC alpha knockout, as you can see here compared to here, and is partly reverted in the NLRP1 deficient zebrafish. So all in all, in this study that just got accepted, we could show that the environmental exposure to Vulcanodynium rugosum might be actually the cause of the dermatitis observed in the fishermen, and that this is probably due to the release of the toxin portimine from this environmental dinoflagellate that will induce a ribotoxic stress response driving to NLRP1 inflammasome activation. Also, we are not equals. Some people actually might be more resistant to portimine due to NLRP1 polymorphism on the phosphorylation side. Finally, I would like to finish on unpublished results to show to which extent studies on the NLRP1 inflammasome in humans might actually contribute to understanding some variabilities between rodent and cell lines. So here, given the high sensitivity of NLRP1 to phosphorylation, driven by ZAC alpha and P38, we hypothesized that additional kinases might be at play also to phosphorylate NLRP1. So what we did is that we used ZAC alpha knockout cell reporter cells and exposed them to a bank of phosphatase inhibitors to see if we could unveil additional kinases. We got beautiful hits by measuring the spec enrichment in our reporter cells, including a full family of toxin and pharmacological inhibitors able to inhibit two major phosphatases in our cells, PP1 and PP2A phosphatase. But the interesting thing for us was also the origin of the toxins. For instance, the dinophysic toxin and the ocadaic acid are coming from the sea, like portimine. And they are well known to induce digestive effect, lung effect, but also skin and eye burn. Another one was the cantharidin. Cantharidin is a toxin that is coming from the beetles, and it's able, as you can see here, to induce very strong necrotic damages on the skin. But cantharidin, given this potency, has been also developed in a Wicant cream that is able to kill the mus molluscum, which is a virus in the skin that you get when your clothes are wet or when the towels are wet. And you get this kind of profile. And with a pipette, you come with a cantharidin and you come to burn these warts. But for many years, it has not been described and known how cantharidin was able to burn. We know that it burns. We know that it inhibits various phosphatases, but the causality has not been shown. So to know how those toxins and inhibitors were able to activate kinases, we performed a PAM gene microarray for kinase activity in response to all of them. And what we could find is that we got various family of kinases strongly activated, including MAP kinases, CDK kinase for the cycle, and also other NF-kappa B associated kinases. To make it short, we have tested all of them, either genetically or pharmacologically. What we could show is that in response to all those toxins, the ZAC alpha knockout responds the same way than the wild type. They do the same specs and activate an inflammasome. The GNK pathway is not involved, or not strongly involved. We have to delete P38 pathway with the 4 isoforms, show a strong reduction, but that is not total in terms of specs. But what we found is that there is an additional kinase that is called TAC1 kinase. And this also induces a partial reduction in the activation of the NLRP1 inflammasome. And this is ZAC alpha independent again. But when we combine P38 deficiency to the TAC1 deficiency, then you all came back to the basal state, suggesting that TAC1, in addition to the P38 kinases, is also able to induce an NLRP1 activation. So at this step, there is plenty of work to do, but what we can conclude from this preliminary result is that the NLRP1 inflammasome activity is regulated through phosphorylation-dephosphorylation mechanism constantly. PP1 and PP2 phosphatase keep the NLRP1 inflammasome inactive in cells, and there are additional MAP kinases involved in its activation, such as P38 and TAC1. Now the aim for us is to understand how the PP1, PP2 can dephosphorylate and promote activation of NLRP1 in a ZAC alpha independent manner, but also if there is a microbial targeting of such a process. Finally, to make an overview on this not so much emerging inflammasome in human, I would like just to first conclude by something that is quite obvious for all of you. Rodents are not human, but I would add also cell lines are not primary cells, because most of the keratinocyte cell lines that people use have lost the expression of the NLRP1 inflammasome, which render those cell lines not responsive to NLRP1 triggers. But also to show you how much it has the catalog of triggers and pathway activating NLRP1 have been expanded, that go from the SARS-CoV-2, chikungunya, HRVs, SFV, viruses, but also various pathogenic bacteria, a lot of environmental triggers, and now various medical and pharmacological tools that are used, including in the aldara cream, the isosteric acid that is able to maintain the cream in addition to the imicream mode. So now many questions about the mechanism of activation of NLRP1, but also the acquisition or the losing of various polymorphism around NLRP1. Are they beneficial, deleterious for the host upon response to all those triggers or individual triggers? Also, NLRP1 is one of the sensors that lack inhibitors. And given the high prevalence of autoinflammatory disease, but also UVB exposure and the emerging of various ribotoxin in the environment, might be interesting to develop NLRP1 inflammasome inhibitors. Finally, one thing that we are still missing in the field of the NLRP1 inflammasome due to the lack of conservation between species is that what is the outcome of the induction of the NLRP1 inflammasome in human. So on that, I would like to thank everyone that has been involved. As you can see, on these three complementary studies, a lot of people, labs, and expertise have been engaged, including the work from Leana, Marco, from the lab, who are in PhD students, from Miriam, who was previously a PhD student and now is postdoc in the Fran Roca lab, but also the fantastic help from Franklin and Kim Robinson, who is now in York, and John Common at ISTAR, from Anthony Elras in Toulouse for the ribosome profiling, the IFREMER, the ERDE, and the University of Dakar on the porcine studies. The private companies Invivogen and Imactive3D. Invivogen has been generating with us all the reporter cell lines for all the inflammasomes, and Imactive have been developing all the software to quantify automatically the specs very easily. And also all the founders, as you can imagine, it has been costing a lot. And of course, in France, do not forget, it's on the 7th March. Thank you for your attention.
Thank you very much, Etienne, for this super comprehensive and very well explained topic. It's a complex topic, but I think you make it actually accessible for everyone, and thank you for that. We have many questions for you. Maybe we can start with one from the audience, from Steve Crampton from J&J. Given the role of NLRP1 in epithelial barrier, do you see potential for targeted therapies that would modulate its activation in conditions such as skin disorder, respiratory diseases, ocular inflammation, etc.?
Definitely, yes. Beyond the autoinflammatory diseases driven by NLRP1, we can see that for the portimine and the environmental exposure or the sunburn, for instance, the accumulation of sunburns due to accumulated UVB, probably dampening NLRP1 or even the kinases that are upstream, ZAC alpha or P38, might be of good help. So yes, there are plenty of opportunities. For now, there is no real fantastic NLRP1 inhibitor like for the NLRP3 field, but yeah, I think it's a good way to go on that. I would like to add also that for NLRP1, it could be also extremely interesting in patients for some diseases, such as the wart induced either by the morculum or by the HSV infection, to have also sensitizer for NLRP1 response in order to remove the warts.
Thank you. And so how common is the NLRP1 polymorphism, and is it associated with more sensitivity to bacterial infection, for example, or more sensitivity to autoimmune disorders, or resistance to portimine, for example?
So For portimine, there is no response because the disease is old of few days, let's say,
Hmm.
officially. But let's say for viral infection, I think it's the Casanova lab in the PNAS paper who showed that some variants of NLRP1 that sensitize to autoimmunity or autoinflammation are actually associated to various viral infection that develop in the skin but might end up in the lung. I think the Setmaster's lab also have shown that some variants might actually be associated with reduction or improving or improvement of the asthma sensitivity in the cohort in Mexico. So you have plenty of this association of NLRP1 polymorphism that have been described in the literature in various clinical and fundamental studies that now can be addressed thanks to all those cell types and tools that have been developed.
Mm. And I was wondering, so the portimine you was found in the net and all around the fish, basically material, but can it be found as well in the fishes and then would be eaten by people and then having, gastrointestinal damages, for example?
Yeah, that was a tricky question for us because usually, when there's toxin, indeed, the fish eat the dinoflagellate and then they have the toxin. We eat the fish or we eat the muscle and then we get diarrhea, basically. Here, nobody got any diarrhea. They
Mm.
got only skin symptoms. And we got the answer is that the portimine at acidic pH is extremely sensitive and it gets degraded. So, which means that we cannot get intoxicated through the gut with portimine. And that's probably why they got mostly intoxicated through the skin and the eye.
That's interesting. And so you nicely show the differences between human and mice, which obviously render in vivo studies very difficult. I had two curious questions. One is, did you try to make a mouse with the same pattern of expression as in human? That's the first one. And the second one, and how do you explain that according, all along the evolution, the NLRP1 has actually switched from the immune to the epithelial system?
So for the first question, I have to confess, Nicola, we failed miserably in the lab. We have tried 3 times. Um, the embryons were highly, highly inflammatory. So the first 2 times we made the mistake to put that on keratin promoters, too strong promoters, I think.
Okay.
So highly inflammatory. We use also the tamoxifen inducible expression for the rodents, and
Hmm.
they also exhibited autoinflammation at the adult stage. So for now, we gave up slightly, but it appears that some have succeeded at having humanized NLRP1 in mice, but not necessarily in the epithelial compartment, which is a kind of a break. Um, so it's a tricky receptor to manipulate. When it is about the second question, I think if you think about Toxoplasma gondii or you think about Bacillus anthracis, they are all activators of the rodent NLRP1, specific NLRP1B in rodent. And those are close to the soil, pathogens that are either in the soil or either the rodents are specific host for those pathogens, such as for Toxoplasma. And it might be that evolution has been driven in those organisms to respond to those kind of infection because rodents are close to the soil or are intermediate or final host of the pathogen. For us, for instance, you take airborne pathogens such as common cold coronavirus, SARS-CoV-2, HRV. Uh, in humans, we all respond to NLRP1, whereas the rodents do not. So probably it's a coevolution between the pathogen and each species according to the physiology and maybe the size of the species.
Thank you. And so you were we had another question regarding development actually. So it's good that you mentioned that the mice did not make it actually because many detection system in human, and now it's known and more in the human immune system, for example, is not only involved in defense of the organism, but it's also involved in the regular homeostasis of of the tissue and also in the developmental process during embryogenesis. So do you think that in human, NLRP1 actually play an important role, either in development or maybe in the protection, at the fetal maternal interface? Do you see NLRP1 in the embryo in human as well?
I never checked because it seems a lot, but we have been working on NLRP1 since only 2021 and the SARS-CoV-2 pandemic, and the major findings from Franklin are only from 2016. So there are plenty of hypotheses that might be relevant right now to address. And you're right, if NLRP1 is expressed in the maternal-fetal barrier, if it's in the vaginal barrier, if it's in some neuronal barrier as well for the brain, those are fantastic questions to address that we have ignored because we did not know how it works. Let me take one quick example. Let's take some neurodegenerative diseases from Parkinson or other diseases. We know that you have accumulation of transcript and translated proteins that can be pathological and affect the ribosome function. So it could well done and well be that in those contexts, I'm not saying that they are responsible, but the ZAC alpha activation due to ribosome inactivation with polyQ, poly-G, or poly whatever chains will lead to an NLRP1 or at least ZAC alpha driven cell death. And that this might be an interesting approach to follow. And regarding the imprinting of NLRP1, this is a dream to me to determine whether it might have a shaping process during the development. Yes.
Very interesting. And also that maybe resonate in terms of drug development, because then if NLRP1, inactivation, molecules need to cross the blood-brain barrier as well. So this is something very interesting for the future. Do you have heard about any NLRP1 deficient humans?
No, not yet. But I think probably they might exist, like Caspase-1 knockouts in humans might exist. Or maybe not, which would mean that NLRP1 is not an essential but almost essential gene, which would be perfect for me. But I'm pretty sure there are deficiencies. But when you see the spectrum of activators and response from the human NLRP1, it could be that actually our cells did not put the eggs in the same basket. And that, for instance, with protein, we showed that there are some polymorphism where the ribotoxic stress response cannot activate NLRP1. But NLRP1 is still responsive to viruses or chemicals that do not go through ribotoxic stress response, which means that there are plenty of mechanisms of action that might lead to NLRP1 and that a full knockout of NLRP1 might not be so much viable for human. But this is just pure speculation at this step.
Yeah. Thank you. And so in terms of sensing, um, two small questions. One is, do you get a dose response system of activation of an LRP1, or is it like an on-off system? As soon as you have activated an LRP1, basically there is no way back. And the second one is that in the same line a little bit, so the toxin that you identified that can activate an LRP1, can they also activate additional sensing factor present on, mammalian cells like TLRs or NLRP3 or any other system?
So I think it's the Dan Bakovkin lab and the Franklin lab who have tried to address what is the threshold, for an LP1 activation. The thing is that this inflammasome is extremely complex because it first needs to be degraded by the proteasome to release the active fragment of this inflammasome, which means that you have still a safety in your cells that will probably be the amount of degradation that occur that will release enough active fragment to promote an inflammasome. And when we have look on the cells, this inflammasome is constitutively degraded, which means that probably we speed up the degradation or we increase the degradation of the regulators or NLRP1 when we activate it. And probably it's a balance between the degraded fragment and the active fragment that make him under regulation. But once it has been launched, you cannot get back.
Okay.
For the second question, We have tested all inflammasomes, all TLRs with sportimine, for instance, with reporter cells everywhere. We have tested also other sensors such as the RIGI, MAVS, MDA5, STING pathway, CGAS, so many. I don't have them all in mind, but basically we did the in vivo gen innate immunity catalog, and there is only an NLRP1. It's terrible. It's like this. It's a ribotoxin. It inactivates the ribosomes and induce this pathway. But if you move out an NLRP1, the ribosome is still inactivated, which means that the ZAC alpha induced P38, GNK, and R pathway are still active, which means that at the transcriptional level, you can very well see the induction of various genes and the compensatory cell deaths that occur.
Okay, maybe a last question before we close the webinar. So you were talking about airway disorders. So let's talk about, for example, asthma. How do you think that NLRP1 is actually involved in this type of frequent disorder? So what could activate them, in the absence of bacterial infection, for example? Allergens, maybe?
That's a good question. I don't have the answer. We have been asking this question in the lab for many years. My hypothesis is that if we rely on what we know for now, and we know that the HRV virus that is associated to asthma exacerbation in human is a trigger of an NLRP1 and can trigger an NLRP1 activation in healthy subjects in airways. We know that when we activate an inflammasome, caspase-1 can actually inactivate one of the major allergic cytokine, that is IL-33, by cleaving it in its active fragment. So it inactivates. So it might be that in some patients they are less responsive to some triggers or some stress, and that this polymorphism make that caspase-1 and maybe caspase-3 are less activated, which means that IL-33 might be way more released in more higher quantity or in more active form than in an NLRP1 without those polymorphisms. But which polymorphisms and how, this remains to be determined experimentally. It's just speculation at this stage.
You were talking about cockroaches or beetles, non-beetles, but
Yeah,
yeah, cockroaches are, I would say, close to beetles in a way that. Do you think that they are one of the major allergens as well in asthma? So yeah, any thoughts about it?
you know,
Not
the
about
thing is that
Beetlejuice or.
exactly, but the cantaridine is extremely strong at penetrating your cells. The extracellular protease, even though they are close to in terms of species of origin, you know, they cannot properly penetrate into your cells. They induce a stress response from the plasma membrane that go inside. And for now, we still need to understand what is going on. One element of response is that uh, previously with Franklin Zhang, we showed that the potassium efflux in epithelial cells is also a trigger of an NLRP1 inflammasome in epithelial cells. So it could be that the stress induced, if I follow you, by those proteases might lead to potassium efflux and NLRP1 activation and or modulation and might regulate this. But it's just pure speculation today.
Thank you. Thank you very much, Etienne. If there is no more further questions, so I would like to thank you again very much for your time, for your nice explanation. I hope that this has sparked a new interest in the industrial and academic community to target NLRP1. I'm sure it is. It is the case. I see many people as well from pharma companies. And you know where to reach Etienne if you need. And again, thank you to everyone for connecting. And I wish you all the best for the rest of the day. Bye bye.
Thanks everyone. Thanks, Nico. Ciao.